But other countries do hold quite significant collections of Pacific specimens. And so in New Zealand, we hold quite a substantial number of specimens held on behalf of some Pacific Island nations. And our collections sort of date back from the early 1930s, but it wasn't until the 1970s when we had a major push to collect a lot of diseases from the Pacific. So in the 1970s decade, we collected over 3,000 different plant disease specimens from throughout the Pacific. This has tailed off a little bit over the decades, but we're still pretty much collecting over 1,000 specimens a decade from the Pacific Islands. In our culture collection here in New Zealand, we also have many hundreds of isolates from the Pacific Islands. And you can see here that there's quite a significant skew, so the Cook Islands is quite overrepresented. And this is because the Cook Islands government funded a couple of our scientists to go over there and very specifically look at, well, not only plant pathogens, but also some native fungi as well. So you can see that some nations are underrepresented or not represented at all in our collection, and they may not even have their own collections. So we've been talking a lot about the importance of getting correct names on pathogens and bringing them up to date. But even without names, fungal barcodes can still be really important. So the problem here is that there's so many unknown fungal species. So for fungi, it's estimated variously between 1 million and 5 million fungal species out there. And for bacteria, I mean, you just pick a number, nobody even really has an idea. The problem is that, like I said with my Colletotrichum description, these descriptions and revisions can take many, many years to resolve. So do you just wait, or can you do something now? So our Biosecurity Agency in New Zealand, EMPI, they barcoded all of this particular genus. That's again this fungus that has two names for the same thing, INFORMATICS and diporthene. They coded them all from New Zealand, from our comprehensive collection. Now, only some of these could actually be matched to a name. This didn't really matter, because what they can do now is whenever they get a new INFORMATICS diporthene disease on a border reception, so something that's come into the country and been held in quarantine, they can barcode that and match it up against their database of what they know is in New Zealand, which is incredibly important. Now, so this happened a few years ago, and now this genus is actually being revised again. And because we've got all this existing data about for the barcodes, we can now put names back onto these pathogens. So in conclusion, plant pathogens in the Pacific are poorly known, even though there's been a good history and lots of people working on it. Just in the major taxonomic revisions in the past 5 years mean that all these historic records really need to be reevaluated. Having the correct names on these pathogens and under the correct modern concept is really important for both biosecurity and trade reasons. So I think that having vouchered specimens in collections, both in the Pacific and other countries, and DNA barcoding these specimens can really help understand the taxonomy of the modern concept of plant pathogens in the Pacific. Thanks for listening. I'll be happy to take any questions.
Thanks very much, Bevan. We're waiting to see if any questions come in on the web. We have 2 minutes. Next speaker is all queued up, I believe. Please use the questions section of your control panel to enter any questions you have for Bevan. In the meantime, I have one, Bevan. There is coordination in the there's a group of 4, I've forgotten the name of it, Canada, the US, New Zealand, Australia, the Quad. Is that it? Could you talk a little bit about the degree to which barcoding is being explored on that by that international group?
Sure. I have sat in one of the Quad sessions, but I'm not a member, so I'm not absolutely up to date. But certainly it is being discussed, and they do believe it's important, and we're trying to get together some coordination to make this happen. There are problems with doing this, and some nations kind of don't, and even internally in New Zealand, some segments like the growers, they kind of don't want you to look. They don't want you to barcode these things because they don't want to know that they've got a new disease in here. But really, at that high level, the Quad level, we realise that collaboration is the way forward, and if we all work together in sort of using these modern names, it's really going to be the best for everyone.
No, I was double muted. Hector Aguilar is asking, what action is taken when a fungi is barcoded but there's no match?
Well, that's the interesting thing. So where I really like, because I'm a fungal taxonomist, and so if there's a bit of an echo in the room there, yeah, thanks. So it is interesting, and so that echoes. If it is coming from overseas and coming in, and it doesn't match everything, then quite likely that shipment won't be allowed in. It'll be treated and then retested to make sure that the fungal disease is no longer present.
Okay, we have another question. Have you reevaluated historical records? That's from Diego Perico. Diego, you might need to clarify that. What do you mean by the historical records and reevaluating? While I'm waiting for clarification from Diego, Lucy Tran Nguyen writes, thanks Bevan, a comment. We recently did some sequencing and barcoding for a bacterial genus, and a vouchered specimen turned out to be something else. So something to note for going through collections and gathering genetic data. So apparently vouchers sometimes have problems that are uncovered when you're doing unknowns.
Yes, that is a major problem. I just looked at a particular bacterial genus called Lactobacterium, which causes soft rots on potatoes. And so we screened all of the ICMP collections I mentioned for the moths, but amazingly only half were actually the Lactobacterium genus. And this is probably a worst case scenario just because of the way that these soft rocks are incredibly hard to isolate the original pathogen from. So yes, absolutely, voucher specimens can have problems, but if possible, always try to include a type specimen in there which I hope should be correct.
Okay. I don't have further clarification from Diego. Bevan, do you want to comment on his question? Have you reevaluated historical records?
We have, and we do try to go back and sequence historical specimens. And from culture collections, this is fine because you can load them up as much as you like and extract the DNA. But with derived specimens, we face the same problems as other collections do, in that specimens over about 30 or 40 years, it's really difficult to isolate the DNA from. So that does present a problem.